Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CUET question paper for the subject zoology conducted in the year 2024. If you are new to my channel, please do like, share and subscribe. If you want CUET question papers of previous years exams for other subjects also, you can go to my channel playlist, that is you can go to my channel Omni Bio Entrance. In the playlist C for CUET exam, there you will find. So coming to the first question. Sickle cell anemia is a disease characterized by patients' erythrocytes having characteristic sickle or crescent shape. Select the correct statement about the disease. So sickle cell anemia, it is a point mutation in the hemoglobin B gene that codes for beta globin subunit of hemoglobin. The mutation, it is due to substitution. So here glutamic acid at position 6 of the beta globin chain is substituted by valine and this results in red blood cells having a sickle or a crescent shape. So first statement, deletion of glutamic acid from the 6th position of beta chain, it is not deletion, it is substitution. So that is not correct. Substitution of hydrophobic residue by a polar one. Here I told you glutamic acid is substituted by valine. Glutamic acid is a polar amino acid and valine is a hydrophobic amino acid. So substitution of hydrophobic residue by polar one is not correct. It should be the other way around. That is substitution of polar residue by a hydrophobic one. So that is seen in option 3. So statement 3 is correct. Substitution of valine residue by glutamic acid 1, that is also not correct. It is substitution of glutamic acid by valine. Then next one. They have given the pKa values and the amino acids. So certain pKa values you have to know by heart. So for alpha carboxyl group of an amino acid, it will be around 1.8 to 2.9. For alpha amino group of an amino acid, it will be 8.8 to 10.8. So A1, B2, that is there in option 1 and 2, 3 and 4 we can not take into consideration. Okay, now C3. Acidic amino acid, aspartic acid. So aspartic acid is having a side chain which is carboxylic acid. If you remember the structure, be thorough with the structure of all amino acids. All the 20 you have to be thorough. So it is having a side chain COH group. So the pKa value for the side chain group of aspartic acid will be 3.9. It is carboxylic acid. So it is acidic. So the pKa value will be 3.9. So C4, that is there in option 2. So option 2 must be your answer. Basic amino acid, that is arginine. So it is having a side chain which is amino group. So it will have 12.5 pKa. So D3. So the answer is option 2. Then next one. Gluconeogenesis synthesizes glucose from non-carbohydrate precursors. Yes, that is correct. So you have to choose the correct answer from the options given below. The main site of gluconeogenesis is the liver. Yes, that is correct. Far lesser extent of gluconeogenesis occur in the kidneys. That is also correct. Very little gluconeogenesis occur in the brain or muscle. That is correct. The second main site of gluconeogenesis is the heart. No. The first main site is liver, and after the liver it is the kidney cortex, that is the secondary site or the second main site of gluconeogenesis. So D is not correct. So answer here is option A, B, C, that is option 2. Okay. Then next one. Again match the following. That is the Drosophila, okay, organism with the number of chromosomes. So these are certain organisms, they are okay, which you have to remember for knowing the diploid and the haploid number of chromosome in them. You have to know that by heart. So fruit fly. Fruit fly Drosophila melanogaster, the diploid number will be 8. So A4. A4 is there. Okay, if you know A4, then you can happily go for option 3. That is the right answer. A4 is there only in option 3. Then nematode, that is Caenorhabditis elegans. It is having a haploid chromosome number of 6. Humans haploid chromosome number is 23, diploid is 46. Mouse, it is again diploid number is 4. So the answer is option 3. Then next one. People with hemophilia are unable to produce a factor needed for blood clotting. The cuts, bruises and wounds of the hemophilics continue to bleed, and if not stopped by transfusion with clotting factor can cause death. Okay, you all know that. So select the correct statement about hemophilia. The principal type of hemophilia in humans is due to recessive X-linked mutation. Yes, that is correct. Hemophilia is a X-linked recessive X-linked mutation. That is correct. These males have inherited the mutation from their heterozygous mothers. Yes, heterozygous mothers means carrier mothers. So one of their X chromosome will be carrying the hemophilic gene. The other X chromosome will not have the hemophilic gene. So these mothers, they have 50% chance of passing it to the son. So that is correct. Fathers have X and Y chromosome, but they don't transfer the X chromosome to sons. Only the females give their X chromosome to the sons. Fathers supply the Y chromosome, not the X chromosome. Then affected males never transmit the mutant allele to their son. That is also correct. I told you males are hemizygous. That is, they have X and Y chromosome. They pass on only the Y chromosome to their sons. So that is correct. Nearly all the individuals who have hemophilia are male. Yes, that is also correct. This is X-linked recessive inheritance pattern. So in X-linked recessive inheritance pattern, the disease condition will be expressed more in the males than the females. So A, B, C, D are correct. So answer is option 3. Then next one. Read the following structure which is a combination of. So here Down syndrome, 47 chromosomes will be there. They have given the karyotype. Then karyotype is 47 plus 13. It is not 13, it should be plus 21. Because Down syndrome is trisomy of the 21st chromosome. Then chromosome formula is 2n plus 1, that is correct. Estimated frequency at birth is 1 by 20,000. It is not 1 by 20,000, it is 1 by 2000 for Down syndrome. Then Part2 syndrome, again the karyotype is 47 plus 21. It is not 21 in case of Part2 syndrome. It is 13 plus 13 it should be. Part2 syndrome is trisomy of the 13th chromosome. And 2n plus 1 chromosome formula is correct. 1 by 2000 is the estimated frequency at birth. No, it is 1 by 20,000. So again A is not correct. B is also not correct. Then Edwards syndrome. Edwards syndrome karyotype is 14 plus 18. That is correct. Edwards syndrome is due to trisomy of the 18th chromosome. Chromosome formula is 2n plus 1. And estimated frequency at birth is 1 by 8000. That's also correct. Klinefelter syndrome, again 47 is the chromosome number. XXY. Yes, that is also correct. The males will have an extra X chromosome that results in Klinefelter syndrome. 2n plus 1 is the chromosome formula, yes, and 1 by 500 male birth. That is also correct. So option C and D are correct. That makes your option as 2. 2 is the correct answer. Then next one. Alcohol enzyme and the type of reaction they catalyze. So alcohol dehydrogenase, it is an enzyme that catalyzes the oxidation of ethanol to acetaldehyde, and it uses NAD plus as an electron acceptor. Okay, so alcohol dehydrogenase, it will transfer electrons from ethanol to NAD plus, resulting in the reduction of NAD plus to NADH. So alcohol dehydrogenase, it is transfer of electrons. So A2. A2 is there only in option 3. So that must be your answer. Okay, we'll see B3. Trypsin. Trypsin, it is a proteolytic enzyme. It breaks down proteins into peptides or amino acids. It cleaves the bond between lysine and arginine amino acids. So it is a hydrolysis reaction. So B3. B3 is there. Okay, that's correct. Then hexokinase. Hexokinase, it that catalyzes the transfer of phosphate group from ATP to glucose, resulting in the formation of glucose 6-phosphate. So it is hexokinase that is involved in transfer of functional group. So C4. Then pyruvate carboxylase. Pyruvate carboxylase catalyzes conversion of pyruvate to oxaloacetate using ATP and bicarbonate. So it reaction catalyzed is bond formation coupled to ATP hydrolysis. D1. So 3 is correct. Then next one, you have to match the coenzyme with the precursor. So coenzyme A, the precursor is pantothenic acid. So A2. Okay, A2 is there only in option 3. See, if you know one, then one match the following which is correct, then you can happily go for, you not even read the others also. It is like that. The options are like that. So A2, so that makes option 3 as the correct answer. But still we have to read and see. You have to read and see for your exam. FAD, FMN will be vitamin B2, that is riboflavin, flavin adenine dinucleotide, flavin mononucleotide. So in the name itself flavin is there, so it is vitamin B2 which is riboflavin. So B3. NAD plus, NADP plus, NAD D plus, that is nicotinamide adenine dinucleotide, nicotinamide adenine dinucleotide phosphate. So it is C will be niacin, that is C4, and thymine pyrophosphate, it is vitamin B1. So the answer here is option 3. Then next one, vitamin precursor and the deficiency disease it causes. So vitamin B1, it is causing beriberi. A2. See here also, if you know the first one, then you can happily go for option 2 as the answer. Then vitamin B2, it is growth retardation. Yes, vitamin B2 deficiency can also promote growth retardation because riboflavin, it plays a role in energy production also. So chances of then cell growth and development also are helped by vitamin B2. So vitamin B2 deficiency can result in growth retardation. So B3. Then vitamin B6. Vitamin B6, it is pyridoxine and it causes dermatitis. So C4. C4 is correct. Then vitamin B12 cyanocobalamin deficiency causes pernicious anemia. D1. So A2, B3, C4, D1. Then next one. Which of the following statements are correct regarding immunoglobulin A? IgA is the main class of antibody in secretion such as tears and saliva. 100% correct. So that statement is correct. IgA is also found in secretions of lungs and intestine. That is also correct. IgA is the first line of defense against infection at this site. Yes, at this, that is tears, saliva, lungs, intestine. It is the first line of defense, so that is also correct. Then IgA is the main immunoglobulin in the bloodstream late in the primary immune response. So primary immune response, it is by IgM. Late in the primary immune response, they might be meaning secondary immune response, and secondary immune response is by IgG. So D is not correct. So A, B, C is the answer. Answer is option 2. Then next one, pollutant and the characteristic. So sulfur dioxide, it is colorless, heavy, water soluble gas with pungent odor. A2. Oh, again, A2 is there only in option 3. Then nitrogen dioxide, it is reddish brown gas, slightly water soluble. Ozone is pale blue gas, water soluble, unstable and sweetish odor. So C4. And hydrogen sulfide, it is colorless gas with an offensive rotten egg odor, slightly water soluble. So 3 is the answer. Then identify the rule that suggests that endothermic animals from cold climates tend to have shorter extremities, that is ears and limbs, compared to animals from warmer climate. So here it is telling about extremities of the animal. So naturally it is Allen rule. Whereas Bergman rule, it is for the whole animal. Okay. So Bergman rule, it states that body size increases with colder climates. So the answer is option 2, that is Allen rule. Then bottleneck phenomenon, it is the reduction in population size due to environmental or genetic factors. And survival of the fittest is those individuals with favorable characteristics are more likely to survive and reproduce. That is in case of your natural selection. So that's it. Then next one. They have given statistical terminology and their definition. So median, the reciprocal of the average of reciprocal, no, median, the middle value it should be. The value of the middle term of series when it is arranged in ascending order of magnitude. So A2 is there in option 2 only. So 2 must be the answer. Okay, mean is the average. So see third one. The value we get by dividing the total of the values of various given items in a series by the total number of items. So you add up all the values that is given in the series and divide it with the total number of items, that is your mean. So B3. Mode is most frequently occurring one. So the most commonly or frequently occurring value in a series, that is mode. And harmonic mean is it is reciprocal of the average of reciprocals of the values of the terms of a series. So A2, B3, C4 and D1 is the answer. So that's option 2. Then calculate the harmonic mean of the numbers 5, 4, 5, 10 collected in one experiment. So harmonic mean is reciprocal of the mean of the values or mean of the reciprocal of the values. That is what we have understood. So harmonic mean is n by sigma 1 by x, where n is the number of values, x is individual values, 1 by x is sum of the reciprocal of individual values. So 1 by 4 is 0.25, 1 by 5 is 0.2 and 1 by 10 is 0.1. Sum of the reciprocals means it is 0.55. Harmonic mean is 3 by 0.55. You will get it as 5.45. So the answer here is option 2. Then following are a few statements with reference to biostatistics. Choose the ones which are incorrect. Not the correct one, you have to choose the one which are incorrect. So type 2 error is alpha error. No, type 2 error is actually beta error. So that statement is not correct. So that is your answer. Then type 1 error is beta error. No, type 1 error is alpha error. So again the statement is your answer. Correct null hypothesis is rejected in beta error. Correct null hypothesis is rejected in type 1 error, and I told you type 1 error is alpha error. So here it is again your answer. Wrong null hypothesis is accepted in a alpha error. No, wrong null hypothesis is accepted in type 2 error, and a type 2 error is your beta error. So again D is your answer. So A, B, C, D is the answer. Answer is option 3. Then next one. After electrophoresis, the gel containing the separated protein is immersed in an acid alcoholic solution of the dye. Okay. denatures the protein. Yes, but actually when you immerse it in acid alcoholic solution, you are mainly fixing it. But there are chances that it can help to denature proteins because alcohols can disrupt the protein structure. Okay, so denatures the protein is also correct. Fixes them in the gel, that is more correct actually. Oh, A, B is correct, A is correct. Ensures they do not wash out, that is what is meant by fixing. Allows the dye to bind to them, that is also correct. So A, B, C, D is the right answer. Then next one. In the process of protein sequencing, trypsin and chymotrypsin are used for generating fragments. Select the tryptic peptide from the list. Okay. Now trypsin, I told you, it cleaves the peptide bond on the carboxyl side of lysine and arginine amino acids. Okay. Lysine and arginine only. Chymotrypsin, it cleaves the peptide bond on the carboxyl side of aromatic amino acids such as phenylalanine, tyrosine and tryptophan. So here they have asked only for tryptic peptide. So out of these options, lysine is there in option 4 only. The others are cleaved by chymotrypsin. So answer is option 4. Okay, which of the following statements related to transcription in bacteria and eukaryotes are correct? During concurrent promoter sequence, recognition and melting, melting commences with base flipping where two bases are flipped out into pockets of the primary sigma factor. Yes, that statement is correct. So this happens during promoter recognition by the RNA polymerase sigma factor. So what is this melting? Melting is nothing but the separation of the two strands of DNA allowing and resulting in the formation of single strands, and this result in creation of a transcription bubble which allows the RNA polymerase to access the DNA template and start synthesizing mRNA. Okay, that is what is melting. Now what is base flipping? So base flipping is single nucleotide base within the DNA strand. It will temporarily rotate out of the double helix and this process it will allow RNA polymerase to access the DNA and help in transcription. So this occurs only with a single nucleotide base. So that is also correct. So A is correct. Then binding of alpha amanitin to RNA polymerase 2 permits entry of nucleotides into RNA polymerase active site and synthesis of RNA but prevents translocation. That's correct. Now you might have learned that alpha amanitin, it inhibits RNA polymerase. The mechanism by which it inhibits RNA polymerase 2 you have to know. That is what is meant with this statement in this question. So what is this translocation coming under transcription? You might have learned translocation in case of protein synthesis, that is your translation. But what is it happening in transcription? So in transcription, it is the stepwise movement of RNA polymerase along the DNA template, adding new nucleotides and extension and help in the elongation of the mRNA chain. That is translocation in case of transcription. So it is the movement of RNA polymerase along the DNA template, adding nucleotides, resulting in elongation of the mRNA chain. That is translocation. So how does alpha amanitin work? It binds to RNA polymerase 2 permits entry of nucleotides into RNA polymerase 2 active site and synthesis of RNA, but it prevents translocation. And alpha amanitin, just to remind you, it is a toxin obtained from mushroom Amanita phalloides. And it inhibits polymerase 2, RNA polymerase 2. This is RNA polymerase 2 is the one which is involved in transcription in eukaryotes. So B statement is correct. RNA polymerase 1 cannot bind to upstream promoters. That is not correct. Then next one, FACT. FACT is facilitates chromatin transcription. Facilitates chromatin transcription. Is associated with the RNA polymerase during transcriptional elongation and helps displace one histone octamers during transcription. Yes, that is correct. So FACT, it is involved in transcriptional elongation and associated with RNA polymerase 2, and it facilitates movement through chromatin by displacing the histone octamers during the transcription. And the histone, that is H2A, H2B dimers, are the ones which are mainly involved here. So D is also correct. So be thorough with your transcription and translation. A, B, D is the answer. Answer is option 1. Then next one, match the following: progesterone and site of hormone and the site of synthesis. So progesterone, corpus luteum, A2. Okay, again also is there only in option 3. So that is the correct answer. B, testosterone, testosterone testis. B3, estrone, ovary. E4, aldosterone, adrenal cortex. This was a simple match the following. So A3 is the answer. Then next one. In the process of exploring eukaryotic ATS ribosome, the following components may be observed. Okay. Choose the correct answer from the options given below. So ATS ribosome, there is two subunits, that is 60S and the 40S. The 60S subunit has 5S rRNA, 5.8S rRNA, 28S rRNA and approximately 49 proteins. Whereas the 40S subunit, it is having 18S rRNA and approximately 33 to 34 proteins. Now you have to choose the correct option. So A, 28S rRNA, 5.8S rRNA. So there is 28S rRNA, 5.8S rRNA in your 60S subunit. So A is correct. 5S rRNA and 34 protein. 5S rRNA is there in 60S subunit and it has 49 proteins, not 34 proteins. So B is wrong. 23S rRNA, it is found in the prokaryotic ribosomes. That is in the large subunit, that is 50S subunit of your prokaryotic ribosome, you will find 23S rRNA. It is not there in eukaryotes. Here the question is for eukaryote. Then next one, 5S rRNA and 49 proteins. So 60S subunit, if you see, it is having 5S rRNA, 49 proteins. So D is also correct. So B and C are not correct. So A and D only, that makes answer 1 as the right option. Then next one, in an experiment, 2 fly populations are separately maintained for many generations. Population A contains closely related individuals, whereas population B contains set of unrelated individuals. Okay, so A is closely related, B is unrelated. Over many generations, it was observed that the fitness of the individual in population A is lower. Naturally, it will be lower because it is having closely related individual, and fitness of those in population B because close relatives will not mate with one another. That is not correct. There can be mating between close relatives also. So A is not correct. Close relatives compete for common resources more than unrelated individuals. Nothing like that between close relatives and unrelated individuals. Whether they are close relatives or whether they are unrelated individuals, they compete for common resources. So 2 is also not correct. The offspring of close relatives harbor more deleterious recessive mutations. Yes, that is correct, because they will have inbreeding. Close relative means it is inbreeding. So naturally there will be reduced genetic diversity and they will be more vulnerable to diseases, and even there will be increased expression of these deleterious recessive mutations, and all these conditions can lower the fitness of population A. So 3 is the right answer. Fourth one, unrelated individuals are likely to contain new high fitness gene. That is also not correct. Then next one, papain digestion of IgG antibody molecule result in. So this is your antibody. It is subjected to pepsin digestion, mercaptoethanol reduction and papain digestion. These you have to remember. That is why I have put that diagram here. So pepsin digestion, you will get FAB-2 fragments plus FC fragments. So these are this FAB2 dash, it is a Fab-like fragment and it can bind antigen. But FC fragment, they are usually not recovered during pepsin digestion because they have been digested into multiple fragments. Then upon papain digestion, you get 2 Fab fragments and 1 FC fragment. Then mercaptoethanol, it will reduce the disulfide bonds, that is why SH here, and you will get this will break, bond will break and this bond will break, so you will get 2 heavy chains and 2 light chains. That is with mercaptoethanol reduction. So be thorough with this treatment of antibody with various agents. So here they have asked for papain digestion. I told you papain digestion will give you 2 Fab fragments which can bind antibody and a 1 FC fragment. So option 1 is the answer. Then select the plant or ecosystem known to have highest energy conservation efficiency. So here it is sugarcane they have given in the option, and I think that is because sugarcane is a C4 plant. And upon checking, I found that it is more energy conservation efficiency than corn and open sea. Okay, so answer is option 2. Then select the vegetation type having highest productivity from the following sites. So desert, it is less availability of water and low vegetation. So that is not correct. Deciduous forest, there is seasonal leaf loss. So there is what is that? Highest productivity is not possible. Then coniferous forest, again slow growth rate and high respiration. So out of these 4 is the answer, that is grassland. Then which one of the following are correct characteristic features of bony fishes? It is homocircle caudal fin, option 4 if you see, that is the one that is correct. 4 pair of gill slits and presence of operculum. So answer is option 4. So that's it. Thank you for watching my channel. If you want still previous year question papers of CUCET, you can go to my channel Omni Bio Entrance. In the channel playlist, see for CUCET exam, and there you will find. Thank you.